All right. Hi, everyone, and welcome. Welcome to the MedTech Talent Accelerators ExploreMedTech seminar. Today's seminar is about technologies in my practice with Dr. Claudia dos Santos. Before we get started, we'll do a land acknowledgement. So if you can give me a second, we can get that started. So just to recognize, I'm here located in Toronto. So Toronto is in the Dish with One Spoon territory. The Dish with One Spoon is a treaty between the Anishinaabes, Mississaugas, and the Haudenosaunee that bound them to share the territory and protect the land. Subsequent Indigenous nations and peoples, Europeans, and all newcomers have been invited into this treaty in the spirit of peace, friendship, and respect. And we hope to carry ourselves that way as we deliver our programming here in the Toronto region. So with that, so again, the idea of our seminars today for the MedTech Talent Accelerator and this is that we really want to learn about clinicians' use of medical technologies, their interactions and engagements with medical technologies in their practice or otherwise, and the initiatives they're involved in. So with that, I'll really pass it on to Dr. Claudia dos Santos. If you could kind of give a brief introduction to yourself, and the floor is yours, Dr. Santos.
Thank you very much. I'd like to thank everyone for the invitation, a very kind invitation to speak to this illustrious group of individuals who hopefully will change the world. So this is pretty exciting. I would also like to take a brief moment to acknowledge to do the land acknowledgement according to the University of Toronto, where I have the privilege to practice, which is traditional land of the Huron-Wendat, the Seneca, and most recently the Mississaugas of the Credit Valley. I have prepared a talk for you guys. I should say I have no commercial disclosures with this talk, and briefly, I want to tell you a little bit about my practice with a focus on my research. I am a clinician scientist. I work at St. Michael's Hospital, and I have a basic research laboratory where I do a lot of translational research. I have prepared these slides, but I have a sense that you are all muted and you might not interrupt me, but I don't mind if you do. So if you feel like chatting in the midst of this, please feel free to interrupt me. I want to talk a little bit about the coronavirus pandemic that we've experienced, severe acute respiratory distress syndrome, and some of the challenges that we have faced, and how we have engaged our collaborators at the National Research Council of Canada, and how we are developing novel technology that we hope will change the way in which we live through pandemics, this one as well as future pandemics. So as you know, the COVID pandemic has had an enormous impact on the way in which we view medical care, not only in terms of the burden of illness and the significant amount of suffering, but also it has highlighted significant weaknesses in the way in which we deliver healthcare. One of the issues that has been incredibly dramatic, and I don't think that I'm exaggerating when I say this, is the concept of time, and how the pandemic really underscored for many of us, especially in the beginning of the pandemic, how difficult it was to obtain critical information in a timely fashion. When I first began in the intensive care unit, working in the intensive care unit, it took us at least 48 hours to confirm that a patient had COVID. That delay of time meant that a lot of us had to be wearing full personal protection. Everything had to be, terminally cleaned between one patient and the other. The response to the pandemic, even in my home country in Brazil, was so dramatic that in the second wave, you saw all those horrible images on TV of patients not having access to oxygen. So triaging of patients, appropriately stratifying patients to proper care, finding out who was going to need an ICU bed, making sure that people had appropriate access to medication, intubation, life support units, mechanical ventilation, oxygen supply. All of this was markedly delayed by our inability to do accurate and fast diagnosis of the virus, which is a problem that sits right in the hands of a lot of biotechnologists of becoming more, developing the technology that is required for fast diagnosis. The other really important thing is the issue of access, that we suddenly realized that being able to diagnose patients in remote locations or locations with difficult access, and being able to use technologies that could appropriately stratify or identify those patients that were going to become really sick and separate them from those patients who were not going to be really sick was really important, and significantly delayed our ability to provide care in more difficult to access areas, or patients that were isolated and not in the mainstream of medical care. The other thing that is, we know for sure that pathogen identification is of critical importance. And in the beginning of the pandemic, you may or may not remember this, but some of the diagnostic tests had 20% false negative rates. So that was impossibly difficult to manage, because we didn't know who was going to be falsely positive. Or falsely negative. I work in the ICU. I'm an intensive care doctor. My patients developed severe acute COVID. Many of them had to be transported to other hospitals for extracorporeal membrane oxygenation. Many of them required very advanced mechanical ventilation, including proning, where we actually ventilate patients upside down or with their face downward, so that we can actually access some areas of the lung. The other problem with acute COVID is that not only was acute COVID a problem, we also had a significant problem now with the long COVID. So patients who, even though they were upfront asymptomatic, are now developing long-term consequences of COVID that affect a variety of different organ systems, such as mental health, the skin disorders, even the ongoing problems with their respiratory system. And we still don't have methods to identify those patients that are going to be developing long COVID, so that we can intervene early in the course of their illness. So as we were thinking about COVID over the past 18 months, what has become really apparent is that while diagnosing the pathogen is super important, understanding the immunological response of the host, so developing those molecular markers that are able to identify those patients that are either going to become severely ill with COVID acutely, or who are going to develop significant and important sequela of COVID, has become a priority in the field. Is there data to support this concept? There is. And there's a lot of data to support the utility of both prognostic and predictive enrichment. So prognostic enrichment is identifying those individuals that are actually sick with the disease of interest, while predictive enrichment is trying to identify those individuals that are more likely to respond to specific treatments for this disease. So for example, in the field of acute respiratory distress syndrome, there are some very compelling data that suggests that positive pressure ventilation using positive expiratory pressure, so not allowing the patient to completely exhale at the end of exhalation, may be good for some patients, while not so good for other patients. So for example, there's some data that suggests that within a cohort of patients that have ARDS, there are two subphenotypes of patients: those that actually have very low mortality and are going to do well with very minor ventilatory pressures, might actually be made worse by the use of aggressive ventilatory strategies. And this has sort of been further validated in external cohorts of randomized clinical trial using specific positive and expiratory pressure strategies for mechanical ventilation, where certain patients that have a specific phenotype that is not always overtly obvious may actually do worse with high positive and expiratory pressure versus other patients who really do require very high end expiratory pressures. So this suggests that in a cohort of patients, there are a subgroup of patients that respond differently to therapy. And we've seen this very clearly with the interleukin-6 antagonist, so the antibody against interleukin-6. So we've seen patients with COVID that have very high levels of interleukin-6. And the interesting part is when we used the tocilizumab, which is the antibody against interleukin-6, to reduce the inflammation in these patients, we've seen some very compelling data suggesting that tocilizumab doesn't actually change mortality in those patients. So being able to appropriately align patients with their therapy is really a challenge for the future. In some subgroup analysis, there may be certain subgroups of patients that actually do well with the interleukin-6 antagonists. And in the field of COVID, this has become a little bit of a holy grail, trying to identify those patients that have different immunophenotypes. So individuals that may have features of high TNF alpha or certain cytokines upregulation, or patients that have immunosuppressed phenotypes, of patients that have the likelihood of actually not responding very well to corticosteroids. And a lot of groups have been using genomic technology, such as RNA transcriptomics, microRNA transcriptomics, and even sort of single cell multiomics analysis to try and tease out from a cohort of patients what is the flavor for each patient. And specifically, this has met with some degree of success with some individuals, some individual groups having shown that there may be some patients that actually have autoantibodies against interferons, which are critical mediators in the antiviral response. The challenge here, though, is that when trying to understand patient responses, it's really important to use appropriate controls. So one of the things that we have contributed to the field is that we have looked at patients who have severe COVID and severe ARDS, but are non-COVID. And very importantly, we've been able to show that even though the literature talks a lot about autoantibodies in COVID, we don't see a difference in autoantibodies between those patients that have severe disease, whether that is COVID or non-COVID, which really implies that the injury, the biophysical injury coming from the mechanical ventilator itself may be actually driving some of this autoantibody production. And this might not really be a feature of the COVID disease. We have been working very hard and trying to identify these longitudinal biomarkers of lung injury and trying to tease out what are the critical features of COVID versus critical features of acute respiratory distress syndrome. And I'm not going to bore you with the design of the study, but suffice it to say that the other really important thing that we find is careful annotation and data management is critical. So a lot of groups are now using machine learning, including our own, and some adventurous groups are also using artificial intelligence to really try to integrate the amount of data and the level of data that we have. This is a major challenge that needs to be addressed from a computational, from a data expertise perspective. Integrating clinical data with multi-omics data, so transcriptomics, MicroRNA omics, single cell RNA sequencing, this analysis of the secretome, so things like metabolomics, proteomics, integrating that data at a patient level is one of the biggest challenges that we have, and will very likely revolutionize knowledge generation in this field. So this is a call for you guys to really think about computational strategies to be able to integrate the level of data. The other thing, of course, is the design of novel and potentially transformative clinical trials such as REMAP-CAP. So this is an adaptive platform trial, and the call for changing the way in which we do clinical research by embarking upon large adaptive trials has really made a difference. And integrating, as I mentioned to you guys, the data from these very large adaptive clinical trials will also be a major challenge moving forward. We've been engaged in another study called the Canadian COVID study, and this is a longitudinal study where we will be following COVID-19 patients for 1 year and generating significant genomics and epigenomics data on these patients so that we can understand the longitudinal response to injury, infection, and inflammation in this cohort. One of the challenges that we have faced moving forward is having the infrastructure, both physical infrastructure in terms of biobanks, as well as the data analysis and data storage infrastructure to collect samples from all of our patients, to annotate those samples, to have systems which allow us to link and analyze data on a very large scale. So one of the efforts that was supported by the University of Toronto, which has really been a game changer for the University of Toronto research community, COVID research community, is the funding of the U of T COVID-19 Biobank, with various scientists leading various groups at key university hospitals, where we are collecting and storing samples and data from our patients, which we hope will then be used for novel knowledge generation. we've randomized already very many patients, and the analysis has already started with about 1,400 patients being analyzed currently. And as I mentioned to you, this has yielded novel information about whether these COVID patients are actually especially susceptible for autoantibody production, and we found that that's not the case. And we have also shown that certain phenotypes of COVID disease are associated with anti-neutrophil titers. So that's also very important. My work has really been in transcriptomics. That's my contribution. We would have loved to collect bronchoalveolar lavage fluid from these patients, but this is another big challenge. This is an infectious virus, so a level 3 facility is required to actually collect those samples. We don't have the bedside containment strategies to be able to collect samples from highly infectious patients in a safe manner that wouldn't put the healthcare providers at risk. So another, I think, thing that moving forward, if we're thinking about being prepared for future pandemics, developing strategies for safe collection and analysis of samples, of infectious samples, is critical. We focused a lot of our attention in blood for obvious reasons, because the blood from COVID patients is non-infectious. Or the blood actually, when put in special tubes that already have RNA lysis buffer, is non-infectious, I should say. We've linked up with a scientist called Bob Hancock at the University of British Columbia, who is a Canada Research Chair in Health Genomics. And Bob has set up a company that he's called CellSep, which is dedicated towards identifying novel biomarkers. And Bob works a lot with machine learning and transcriptomics. Arjun Bagala is his PhD student, and together we have profiled about close to 500 COVID patients, and we have identified that these patients actually fall in 4 phenotypic groups. 4 phenotypic groups which we have named the neutrophil suppressive phenotype, the inflammatory, very high inflammatory phenotype, an immune adaptive immune phenotype, as well as an interferon high interferon phenotype. And each of these subphenotypes of patients are relatively indistinguishable based on clinical data. What they have different is a completely different molecular profile, characterized by a set of genes that are differentially expressed in these patients. And when we looked at the outcomes of these different phenotypes, so this is a survival curve. And what we did was we stratified all the patients that we did the transcriptomic analysis on. We stratified them by their underlying subphenotype, and we looked at their likelihood of mortality. And we were able to show that the phenotypes are not only distinguishable at the molecular level, but they also have very different outcomes. And Bob has identified an 8-gene signature that can be used to profile these patients and assign them into their likelihood of doing well in the ICU or after admission to hospital. We are now working very closely with our collaborators at the National Research Council of Canada to develop a multiplexing strategy so that we can multiplex these host response genes so that they can be used to triage patients at the point of care. So our partners at the National Research Council of Canada are Theodore Veris and Dr. Lydia Malik, and they have developed a completely novel microfluidic portable device that can actually amplify nucleotides, nucleic acids within a few minutes. It's less than 60 minutes, including the RNA extraction at the point of care. The Powerblade device has been patented and actually has been developed together with the Canadian Space Station. Sorry, with the Canadian Space, well, whatever, the Canadian Space Investigators. But it has been approved for deployment in the International Space Station. And it should be, and it has already been deployed in the Concordia in Antarctica. The idea is that this portable device is easy to use, user-friendly, can amplify these markers on any kind of sample from patients. So we've tried this with saliva, with nasopharyngeal swab fluids, as well as with blood, and can allow you to profile patients and identify the immune subphenotype of patients at the point of care. We've then partnered with Axel Gunther and the CRAFT Institute at the University of Toronto. So CRAFT is the Center for Research and Application in Fluidic Technology. And what we've done is we've created at St. Michael's Hospital an outpost of CRAFT. So the outpost of CRAFT really is a translational research station that we've posted inside of the ICU that is ran by our Chief Research Coordinator, Marlene Santos, that is linked to our biobank that is managed by Valeria Di Giovanni, and that where we analyze samples together with Pam Plant. She's the head of the genomic core facility at St. Michael's Hospital. And the idea here is, rather than develop device and deploy devices, you know, traditionally what happens is people do all of the development in their laboratory, and then they bring the finished product to the stakeholders and the users, and they expect the users to adapt to the specifications of the device. So what we're trying to do is actually change that. We're bringing the scientists, so we're bringing the physicists, the clinical chemists, we're bringing the engineers and the bioengineers and the data scientists. We're physically bringing them into the intensive care unit so that they can understand how we work in the intensive care unit, how a nurse works, what is the knowledge base, what is actually needed at the bedside, so that the scientists and the stakeholders can actually develop the technology together. And we hope that this will not only fast track the development, but also help develop a device that is much more in tuned, much easier to use, and it will facilitate deployment of the technology in the field, because the engineers will understand exactly what the challenges are, you know, what the timescales are, how difficult it might be for a nurse or a research coordinator, for a staff physician to actually apply their technology at the point of care. And hopefully that will significantly accelerate the way in which we're doing device as well as biomarker development, because you can well imagine that when we trial all the technology and benchmark the technology at the point of need, we need to be able to do this together with the scientists. So for this reason, we've also set up a basic science benchmarking facility inside St. Mark's, where we have all of the equipment and the resources required to actually benchmark the performance of the technology, the analytical performance of the technology. So again, this is our research station inside of the ICU. So far, from a research perspective, it's worked really well, but we haven't actually trialed the device yet. So we'll see how that goes. So in summary, how we're using technology, we're trying to innovate biomarker discovery in 3D from 3 different aspects. We're trying to discover novel biomarkers that are not the traditional ELISA-based, circulating biomarkers, using composite markers. And we're doing this using a lot of machine learning and integrated clinical data with a lot of validation from external cohorts, so that we can find very robust biomarkers, because at the end of the day, it's garbage in, garbage out, right? You may think your technology is fantastic, but if you're not actually asking the correct thing, it's grossly useless. We're also trying to transform the detection by using very novel microfluidic technology that the material scientists have shown is independent of body fluid, so that we can use any body fluid to actually make detections. And we're also trying to develop a completely new way of biomarker development and technology development by bringing the scientists, the engineers, and the stakeholders together in a physical location, so that they can work together to develop the technology and accelerate advancements. Our team is very large. We have a lot of stakeholders that have contributed a lot of knowledge and enthusiasm to the project. Thank you very much for your attention. I will take any questions.
Thank you. Thank you so much for that, Dr. Santos. So just before we start taking questions, I'm just going to be turning off the recording and the live stream.